All right, our next speaker this morning is Dr. Clawson. Michael Clawson received a PhD degree in microbiology from the University of Connecticut in 1999, where he studied the coevolution of bacterial symbionts known as Frankia. From 1999 to 2002, he did a postdoc at the University of Connecticut Health Center and worked on vaccine development for the human pathogen Babesia microtia. Since 2002, he's been with the USDA ARS as a molecular biologist at the US MARC. His primary field is in the genetics of infectious respiratory disease susceptibility in cattle and sheep. Additionally, he has conducted research on transmissible spongiform encephalopathy, genetic susceptibilities in cattle and sheep, and has developed phylogenetic subtyping and classification methods for small ruminant lentiviruses and Shiga toxin producing E. coli 0157. Welcome.
Thank you. I'd like to thank the organizers for having me here today, and thank all of you for coming, and it's nice to spend some time talking with you today. So I'm going to talk to you about an ongoing project on Mannheimia haemolytica. It's a fairly new project. It's not done, and I'm just going to spend a good portion of the talk trying to build an argument for what we're doing, why it's important, and where the research is going. I'll show you pretty much the latest data that we have, and hopefully you can see that we have some good direction going. Okay. And the other thing I want to do before I start is, I have an acknowledgement slide at the end, but I do want to acknowledge one person in particular. This is a relatively new area of research for me. Why am I doing it? Well, a good reason why I'm doing it is because our group got involved with the Kansas State University group a couple years ago, Mike Apley in particular. And a lot of the questions that this research could address on MHEME are questions that Mike Apley has had for quite some time. And I would like to just acknowledge Mike as the facilitator for this research and for pretty much making a lot of it happen. Okay, so the research that I do in general is on the host-pathogen interaction. And the model there is that genetic variation on either the host or the pathogen side can influence host-pathogen interactions. So some of you are probably saying to yourselves, is this guy, he's got a sheep up there and this is a cattle meeting? And I'm picking on sheep because we have done some work on the host-pathogen interaction in sheep using a lentivirus and stuff going on there. And one thing I'll say is that every now and then I'm going to say to you, this is something I really hope that you'll pay attention to and try and remember amongst all the rambling. And this is one of those things. That when you have a host-pathogen interaction, and you have a population of a pathogen, and you have a population of a host, the question isn't, are there alleles on either the host or pathogen side that are affecting the outcome of that interaction? That's not the question. It's not if. The question, because the answer to that is yes, there is. There are genetics on both the host and pathogen side that are going to affect that interaction. The question is, what is the frequency of those alleles, and what is the order of magnitude of their effect? What is their penetrance? So, in humans are susceptible to the flu. One human might be, we'll call it 1x, has a 1x susceptibility. Another human might have 1.0001x susceptibility. That's different. Is that really useful? Probably not. On the pathogen side, one flu virus versus another, if there's one strain that is 0.0001x less virulent than the other, that's of interest. Bird flu. If you get infected with the bird flu, that's a mortality rate of 90% versus another kind of flu where not quite so much. So, yes, there are effects on both the host and pathogen side. The question is, what are the frequency of alleles and what is the magnitude of the effect? And so the goal of the host pathogen research, interaction research, is to identify genetic variation that impacts health in livestock. So we're working at the nucleotide level. We're defining genetic diversity at the nucleotide level. This is alignment of a gene. There's a lot of variation there. And so a graphical way to make sense of that diversity and to work with it is to make a phylogenetic tree, such as this one here. This is a typical tree you would see for pathogens. Each one of these branches represents a unique sequence that a pathogen has. The sequences all end with the pathogen at the end of the branch. These are all these sequences represent a pathogen with the sequence, with the outer taxonomic unit in the tree. There's no internal nodes on the tree. And what this tree is basically showing is that, in a graphical way, is that, so here you have a cluster of sequences. They are different, but they are clustering together because they have some relatedness. Same is true here, and the same is true to a degree here. These two pathogens have sequences that are related, but they're different enough to have their own unique long branch lengths that suggest that while they're related, they're not quite that similar to each other, as opposed to here where they are very closely related. This is a tree that you might see for an animal. This does allow for internal nodes on the tree. Each one of these circles represents a unique sequence. This is a different kind of tree because when you're making trees for livestock, they are interbreeding, and the ancestral phylotype can be in the population. So this, again, is just a different kind of a tree. But it's basically saying the same thing, that these sequences are very closely related to one another, much different than they are to sequences in this part of the tree. So when you define genetic diversity, you can do it on a bunch of different levels. You can do it by looking at a single gene. You can look at it by looking at genes that are in an operon, or you can sequence the entire genome. And depending on what level of the genome you cover with your sequencing, you're going to have different levels of resolution. And in some cases, you can actually have a different phylogenetics picture because different parts of the genome can have different evolutionary origins. This is a kind of a hard slide to transition to, but this is driving home the point that even when you look at the whole genome of bacteria, for example, not only can they differ in their genetic sequence, but they can differ by the presence or absence of segments of sequence. So what you're looking at is a comparison between two E. coli genomes. K12, which is your common laboratory strain of E. coli, and O157H7 from, it's called the Sakai strain. It caused a massive outbreak in the Sakai prefect of Japan. Any sequence, any bar that's going above the horizon here is a sequence that is unique to the Sakai strain. So all these upward lines here are representing sequence that the Sakai strain has, the K12 strain does not have. And the reverse is true here. These lines down here are representing sequence that K12 has that the Sakai strain doesn't have. So these are both E. coli. The genome size of Sakai is 5.5 megs. The genome size of K12 is 4.6 megs. They only share a 4.1 megabase conserved backbone. This slide also helps me to remind me to say that a lot of these virulence factors, which the O157H7 strain has that makes it virulent, came in with phage infections of the genome. So people get viral infections. Bacteria get viral infections. Bacteria can literally infect bacteria, integrate their DNA into the genome of the bacteria, and give it unique qualities. And in this case, the shikatoxins, which are major virulence factors of S-O157, came in through phages. A locus for enterocyte effacement, which is how this bacteria can bind to epithelial cells, came in the backbone of phages. So this is a form of evolution that bacteria undergo. They aren't static in their genomes. They can do orthologous evolution, where they just have mutations that occur over time. But they can gain and lose large chunks of genomic sequence through things like lateral gene transfer events, virus infections that can give them very unique qualities. And this is how these bacteria can change properties and adapt to their environment as needed. Okay, so going more towards Mannheimia haemolytica, if you want to type strains of M-heme using genomic sequence, basically what you want to do, and what we're trying to do, is you sequence whole genomes, and then you dial into the core genome that all the bacteria in your population seem to have. So you can type those unique phage integration sites. They are useful, but if you want to type bacteria, it's useful to have a system of markers that will work in your population that you're targeting. So here's just a cartoon of 4 strains that have a series of single nucleotide polymorphisms. So each site here is a polymorphism. It's a G or an A. So strain 1 has a G allele at this site. Strain 2 has an A allele at this site. Here's another site where these 3 have a T. This one has a C, and so on. And so these SNPs, that by themselves are not very informative. They just put things into different piles. But when you start to concatenate them, then you get what I'm calling a polymorphism-derived genotype, which is essentially the concatenated SNP allele. So this sequence, GTG, CTG, is coming from here. And when you start to make phylogenetic trees with more and more SNPs that are not linked to one another, you start to pick up some phylogenetic information. So this is a very crude tree that's made from the variations for these 1 strains. You can see that 3 and 4 are clustering close to each other, as are 1 and 2. The reason for that is that strain 1 and 2, they differ by 1 allele only. So here they are on the tree. They're clustering close together. Strains 3 and 4 differ by 1 allele. So they're here. They're clustering close together. But for example, strains 2 and 3 are differing by 4 alleles, which explains why they have all this genetic distance between them on the tree. And so the more nucleotides that you can identify that are informative, and by that I mean they're not linked, the more power you get to start to resolve these different strains from one another. Okay, so I'm going to give you an example of that. This is work that we did do on STEC 0157. This is a work that I did with Jim Bono at the U.S. Meat Animal Research Center. And we sequenced the STEC 0157 genome from 102 cattle and 91 humans. We found a whole bunch of polymorphisms that were not in phage integration sites. And we came up with a test that actually captures alleles for 762 polymorphisms, and we come up with a tree like this. So this is a rooted neighbor joining tree. It's I do this a lot. I always mess around with the pointer. This is a rooted neighbor joining tree. It's rooted with bacteria from which 0157 evolved from, O55. So the root is right here, and then the tree just progresses out. And what it's showing is that there are what we're calling 8 major lineages of STEC 0157. There's 1 here, 1 here, 1 here. Lineage 5 is here, and so on. Now, a lot of you are familiar with STEC 0157. It is a problem in the cattle industry. It's an adulterant. It is considered an adulterant. It's not tolerated in the human food chain. And cattle are a reservoir for STEC 0157, and that's not really up for much dispute. But what this tree is saying is showing a couple of things. One thing is these pie charts are representing the frequency with which we see these genotypes in cattle or in humans. So human are black and cattle are in white. And here we have an entire lineage of STEC 0157, and this is not a rare lineage. These are STEC 0157 you commonly find in cattle. You commonly find them in cattle. You don't commonly find them in diseased humans. This is a whole lineage of STEC 0157 that does not associate with human disease. So one thing this tree is showing is that not all STEC 0157 are the same in terms of their propensity to associate with human disease. And that's an informative thing. Another thing that this tree is actually showing, which I find kind of interesting, is that STEC 055H7 is a human pathogen. It's a common source of infantile diarrhea around the world. If you look for it in cattle, you will find it if you look deep enough, but cattle are not considered to be a reservoir for 055H7. They just aren't. You just don't find it there very often. So the root of the tree is with a human pathogen. The first lineage in the tree is represented by a kind of a special strain of STEC 0157 that are found in Europe. They're found in diseased humans, but you don't really find them very commonly in cattle either. Cattle don't jump into the tree until here, and they go on, and the rest of the tree does associate with cattle. But the base of the tree, the evolutionary base of the tree, are strains that you find in humans and not in cattle. So one thing that this tree is suggesting, other than there are differences in STEC 0157 associated with human disease, is that it's possible, this tree might suggest, where did cattle get STEC 0157 in the beginning? Well, I would argue that this tree is presenting a case that O157H7 may have evolved from O55H7 in humans. And that at some point early on, humans passed it on to cattle. It took off in cattle, and now is passed from cattle back to humans. But in the beginning, it's possible that humans might have given it to cattle. And this lineage here appears to be a lineage that may have evolved away from human virulence. So that's sort of the take-home message there. So if you'll just bear with this example for a second, if you have a cop show up on your doorstep, and for some of us that happens more than others, and he says to you, let's pretend everyone's name is Mike in the room. He says, Mike, you're on 7th Street and you were going 80 miles an hour in a 45-mile-an-hour zone. Oh, and by the way, when you drive a motorcycle, both tires have to be on the ground. I could give you a ticket for speeding, or I could give you a flask of 8 lineages of Aztec O157, and you have to drink one. Well, I know it's one I would drink. It would be, not that I'm recommending anyone do this, it would be this one. I sure as heck would not drink this one, because this is the lineage that contains O157 that is associated with the spinach outbreak of 2006, and this is a lineage that seems to be associated with pronounced human virulence. So the take-home message of this tree, here we have a human pathogen that is not equally pathogenic to people. Okay. So that's very useful. So when you type pathogens by themselves, it's very useful to have a tree that shows their propensity for disease. If you're really lucky and you type pathogens on both the host side and the pathogen side, you're better. And so this is an approach that we try and follow whenever we can. On the pathogen side, you can create a phylogenetic tree of the diversity on the pathogen side, one for the livestock side. And when you have that in place and you have meaningful phenotypes, you can start to test regions of the pathogen tree for increased or decreased virulence, and you can tie that in to regions on the host side that associate with increased or decreased susceptibility to disease, and you can start to see the context in which these associations happen. Whether this decreased virulence is correlating directly with decreased susceptibility, or vice versa for the increased side, and you can start to test. And you can ask a question, do pathogens that have decreased virulence, how do they do with hosts that have, by their genetics, have an increased susceptibility to disease? And perhaps the more interesting one, when you have a host that has decreased susceptibility to disease due to its genetics, how does it fare when it is challenged with the most virulent strain of your pathogen in a population? And again, genetic variation on either side will have an influence. It's a question of allele frequency and penetrance and order of magnitude. I'm going to give you an example of that real quick. This is the ovine progressive pneumonia example. So this is a small ruminant lentivirus that infects sheep and goats. It's related to HIV, although it doesn't affect T cells, so it doesn't have that sort of AIDS phenotype to it that HIV does. But it does cause a pneumonia in sheep, and it can cause them to waste away and eventually die. And there was a discovery that was made at the USB Animal Research Center by Mike Heaton and Craig Glymaster and others, where they tested sheep for, and through a case control study and a genome-wide association study, they tested sheep for resistance to this virus, or decreased susceptibility to this virus. They did a case control with animals that were infected, animals that were not, that had been exposed, case control design. They did a genome scan. They found a locus containing the TN154 membrane in it. It's a gene that's on the outer membrane. I do this a lot. So in this protein, they found that there's an allele, E35, that causes increased susceptibility, and there's an allele, a glutamate, that causes decreased susceptibility in this protein. So on the host side, there's a marker in this protein, TN154. Sheep that have the E35 are more susceptible. Sheep with the K35 are less susceptible. So a couple years ago, a scientist from Brazil came up to my lab, Lucia Sider, and we worked on the pathogen side, and what she showed is that on center there are two subgroups of the small ruminant lentivirus, and that they have an increased affinity for affecting either E35 or K35. So subgroup 2, which is shown here, likes to infect sheep with E35, and subgroup 1, which is here, likes to infect sheep with K35. So there actually is an effect of the strain and the host in terms of the outcome of disease. And when you talk about managing OPP, or this is called ovine progressive pneumonia, in sheep, you have to take into account genetics on the host side and genetics on the pathogen side to know how your alleles are going to work in your flock with the strains that you have present. Okay, I'm not going to belabor this too much because we're running out of time. There are multiple pathogens, and there are multiple, on the virus side, multiple pathogens on the bacteria side, multiple environmental factors that cause BRDC. And a very useful approach for us is to try and dial into one pathogen in particular and test genetics on the pathogen side with genetics on the host side to see if we can flesh out an effect. That for us seems to be a useful thing to do, and that's why when Allison was talking about the study yesterday and about those swabs that are being taken for those animals on the dairy side, that's very important so that you can dial into what genetic effect is protecting against specific agents. Okay, so Mannheimia haemolytica is the principal bacteria isolated from cattle. It's in the Pasteurelliaceae family. It's gram-negative non-motile rod or coccobacilli. It looks like sort of a gray, dull gray colony on a blood plate. And just the background on it, we've known about it since 1885. It was renamed Pasteurella haemolytica in 1932, and then it was renamed M. haemolytica in 1999 based on some phylogenetic analysis that was done. Okay. All right. It is commensal to nasopharynx and tonsils. It gains lung access to the lungs as an opportunist. And I won't go through all these virulence factors. It has a number of virulence factors that essentially allow it to avoid phagocytosis, get the nutrients it needs, bind to the cells that it needs to bind to, and it does have one major virulence factor, the leukotoxin, which this is a toxin that will basically cause leukocytes to burst, causing damage in the lung with, in some cases, fibronecrotic pneumonia developing and in some cases, disease and death. Okay, so M. haemolytica are phenotypically and genetically diverse. This is just a cartoon of a prokaryotic cell. On the left, we have the gram-positive side. On the right, the gram-negative. So for both, on the very outside of these cells, we have a capsule, which is a virulence factor. It does interfere with or prevent these cells from being phagocytized. And these M-heme have been phenotyped by capsular serotyping. And a number of strains are not typable by this method, but there are 12 capsular serotypes that are known here. And of those, there are 2 that are overrepresented in animals that are diseased, the A1 and A6 capsular serotype, and there is another A2 that's commonly found in the upper respiratory tract of animals. It is not found commonly in the lung of diseased animals. And so this suggests that there might be some difference in the types you find in the upper respiratory tract versus the lower. It could be a genetic thing. It could be a phase variation thing. But it does represent that there's a disparaging dichotomy between what you find in the upper respiratory tract as a commensal versus what you find in the lung of a diseased. Very recently, a paper came out in 2004 by Klima et al. In Canadian Journal of Veterinary Research. They looked at M-hemes coming from healthy cattle versus diseased cattle in a feedlot. They found a couple of things. One is they found, again, that the serotype 2 was overrepresented in the healthy. Serotype 1 and 6 were overrepresented in the treated. They did pulse field on those strains. They made a tree from the pulse field. You can make trees from pulse field patterns. Basically, the black is representing cattle that are not treated for BRD, and the white is representing those that are. And you can see that in the pulse field dendrogram, there is a large clustering of black circles here on the right. These are animals that aren't treated for BRD, and you can see their serotypes here. There's a lot of 2s. On this side of the tree, these are cattle that were treated for BRD. A lot of 1s are here. So by pulse field, which is a measure of genetic diversity, we see the same thing that we kind of see with the serotyping, which is that there's a disconnect between what you see in the healthy versus what you see in the disease. So for me, this really did suggest that there is a very strong genetic difference between some of these M-heme strains. So the correlation between phenotypic and genotypic. Okay, so that brings in Mike Apley and his group and some of the questions that they were dealing with and some of the questions that other people in the M-heme field are dealing with. What is the structure of M-heme genetic diversity? What is the distribution of subtypes in cattle? Are some subtypes more virulent than others? And to do that, you do need an improved resolution of M-heme subtypes beyond capture serotyping and pulse field. Pulse field is a very useful tool. It's very, very nice, but we live in an age of sequencing whole genomes now, and you can increase your resolution with through whole genome sequencing and SMP profiling. Okay, so real quickly, there are several keys to success in developing a nucleotide polymorphism-based system for typing strains. One is funding. The other is a biological knowledge of the organism. And this is where I don't claim to be an expert. I claim to be working with experts that provided us with some expertise that we needed. This is very important. You need a deep collection of strains that are separated by time and space. You need to find and characterize the diversity of the population you're interested in. So if we're interested in M-heme in North America, we need to get a sampling of M-heme throughout North America, a representation of that diversity, so that we can make a test that will detect it and characterize it in a way that we can find useful. You don't want to just go out to an outbreak, get 200 strains from that outbreak, sequence all those strains, and say, okay, this is the diversity, this is representation of the diversity. It's not. It's diversity that happened in that outbreak. It's not representation of diversity you might find throughout North America. Okay, the more extensive phenotypic information you have on the strains, the better, because you can hang that information onto your tree to start to make sense of it. You do need a fully assembled reference genome sequence, and you need some good sequencing technology and bioinformatics and support to make sense of all the data you're going to generate. Okay, so we have funding, we have biological knowledge of the organisms for that deep collection of strains. Mike Apley hooked us up with Bob Murray and colleagues at Zoetis. We have a collaboration going on with Zoetis. These guys have been great to work with. It is a collaboration. It's not a patenting, licensing thing. The information that we're going to generate with the strains that we've gotten from Zoetis will be published. The information will be freely available. There's no deal of any kind to make that information not available to the public. Okay, so Zoetis has been running a pathogen surveillance program. They've been collecting strains from all over North America for a number of years. So they were very kind to let us go through that collection and identify strains that would be useful to us because they were epidemiologically unlinked to each other. So there are about 18 universities that had donated strains to that collection that we had to get a transfer agreement with, and we did do that. And we have now two populations. One's a discovery, 158 strains, they represent 35 of the U.S. states, 5 Canadian provinces. They cover 2002 to 2011. Every single strain in this discovery population is epidemiologically unlinked to every other strain in the collection. We have a validation pool, 163 strains, 29 states, 3 Canadian provinces, same time zone, and same deal. Every strain in this collection is unlinked to every other in the collection. There's more beef cattle germplasm represented in this collection than there is in this one. Between the populations, these strains are linked. So within, they are not linked. Between, they are. So this is a total of 321 M-heme lung isolates. These came from diseased animals, dead animals, the lungs of those animals. This collection represents a deep sampling of M-heme strains coming from sick animals throughout North America over the last 10 years. It's a very powerful collection. Then we have another that came through a study that was done with Mike Apley in Kansas State. We have 3 different sale barns in 3 different states. We have these are all upper tract nasopharyngeal isolates, all from healthy animals. These were isolated by non-biased methods on non-selective plates. M-heme, if you grow it on a blood agar plate, it can show a lysis. That can be a tip-off that it's M-heme. We did grow these isolates on these BHI blood plates, but we did not select for any kind of leukotoxin, evidence of leukotoxin, because some strains aren't making toxin enough to see that kind of ring around the colony. We didn't want to have a bias. So there are 73 animals that we have. Strains are collected from 73 animals that have showed no sign of clinical BRDC. Okay, I'm running out of time, so I'll just say that we used a biolog to identify these mHeme as mHeme. It's a phenotypic test. Take your strain, you don't know what it is, you put it on a plate, 96-well plate in this machine. It will do a series of phenotypic measurements, measuring the reaction as the reaction takes place every 20 minutes. It gives you a barcode, and you can use that barcode to tell what your bug is. And the entire collection from Kansas State and from Zoetis has been typed that way. Okay, I'll skip over that in the interest of time. Okay, so we do have a fully assembled genome, which we need to get our typing system in place, thanks to the efforts of Greg Harheit, U.S. Mark, and others who have been doing some full-length genome sequencing. The sequencing technology that we're using is the MiSeq. It's very, very quick, very powerful. In a couple of days, you can literally grow up your bacteria, 96 at a crack. In 2 to 3 days, you can grow your bacteria, make your library, run them on the MiSeq, and get at least 10x coverage, if not much more. Okay, so this is an output from that sequencing here. This is a these are sequences from a single strain that are mapped to a reference sequence. The reference sequence has a G. This reference strain has an A. This is a single nucleotide polymorphism. Analyzing these things one at a time is very easy, very trivial. Analyzing over 1,000 genomes at once, which is what we have in our collection now, is a little bit more of a challenge. And that's what we're using the University of Louisville to help us with. They're developing a pipeline to analyze all 1,000 genomes at once. That pipeline is now in place, came online last week. Analyze all genomes at once. And I think down the road, where we're going with the University of Louisville is that we will set it up so that this is an open website, open for public use. Anyone will be able to deposit sequence. The website will map that sequence to reference genomes, make a tree on the fly so that you won't need someone like me to interpret it for you. You'll just get it through that website. That's the goal. So real quick, I'll just wrap it up. I'll show you a little bit of data on the leukotoxin gene that we've generated. It's a 4-gene operon. There's activating protein, 2 transport proteins, the actual structural protein is leukotoxin A. It's about 2,800 bases. I've just show you, I've looked at some data from 46 clinical lung isolates and 60 non-clinical nasopharyngeal isolates. This is the most uninspiring neighbor joining tree you might ever see from the lung isolates. Here it is. You basically have 44 isolates have identical sequences. One has one SMP different from the other 44, and then there was one isolate that was markedly different from the other. So this tree is basically saying there are 2 clades in the leukotoxin sequence. The 45 46 clinical isolates had highly related leukotoxin sequences, which is pretty remarkable when you look and see where these strains come from. They're coming from Canada, they're coming from the United States. There's 2 Canadian provinces, 12 U.S. states, 10-year span represented. And at this location, you have near clonality. I'm not saying it's all the way through the genome, but I'm saying at this location, you have near clonality in these strains coming from the diseased animals. Okay, so if you make a tree of those with everything that's in the GenBank, so here we have the clinical isolates. Here you have also clinical isolates. When you pull in everything from GenBank, from different species, you see there's 2 sort of clusters. The only thing I'll point out here is that there's very little A2 representation here. This one clinical isolate that does place here is clustered with other A2s. And so it does appear that there's a leukotoxin gene is capturing information that's running along the lines of A2 and A1 and A6. So what about those upper respiratory tract strains? So when we analyze those from 60 cattle, we get 4 clades. 1 here, 1 here, 1 here, 1 here. So there's greater diversity here than what's seen in the 46 isolates from the Zoetis collection. When you analyze them all together, and this is the last data slide I'm going to show, this is the phylogenetic tree. It got so busy, I just collapsed the clades down. So here we have clade 4. There are 0 strains from sick animals, 5 from non-sick. Clade 3, 0 strains from clinical, 27 from non-sick. 2, 1 strain from the clinical, 24 from the non. And here we have clade 1. There are 45 strains from the clinical, and there are 5 strains from the non-clinical that also placed right here. So the bottom line of here is that the takeaway I want to send here is that all M-heme are not all the same. They are genetically different. And when we talk about bacteria that are angry and are ready to move down into the lung, I would say that at the phylogenetic level, it's premature, the story is emerging, but at the phylogenetic level, there appears to be a genetic subtype of M-heme that is angry by nature. It is primed and ready to cause a problem if the circumstances allow it to do so. So where things stand is that right now about 1,000 M-heme genomes have been sequenced. These genomes are being analyzed for SNPs in a newly created bioinformatic pipeline at the University of Louisville. Early analysis does indicate a subset of M-heme causes the majority of disease, which is not a surprise based on the capsular phenotyping and the pulse field data that has been coming out recently. And the question that is coming from this is, will this observation hold up, and can you make a field test to detect pathogenic strains of M-heme? So if you can't treat your cattle metaphylactically, what can you do? Well, one thing you might be able to do is type your cattle for the kinds of pathogens they're carrying. And if there's a small preponderance of cattle that are carrying pathogens with a large propensity to cause disease, perhaps down the road it might be useful to identify those cattle and sort them away from the cattle that don't have those strains and possibly prevent an outbreak from happening. And that's something that just, we'll see if that happens or not. I do want to acknowledge a lot of people that made this work possible, in particular Mike Apley, Brian Lubbers, Robert Larson, Keith Donder, Sarah Kaplik, and Brad White at Kansas State. Like I say, Mike Apley really has facilitated this research. On the Zoetis side, it's Robert Murray, Michael Sweeney, and Ellen Portis. Ted Kauflesh is a bioinformaticist at University of Louisville that is making a pipeline which is really essential for analyzing this data properly. A whole bunch of U.S. marked folks, in particular, Jenny Shula Chavez is a technician who has generated all the libraries you saw and helped generate a lot of the protocols on the fly to make this possible. Okay, thank you.